Hello everybody, and this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And so far what we have discussed about the different discoveries made by the scientists to unreveal the different aspects of photosynthesis. Then subsequently we have also discussed about the light reaction as well as the dark reactions. So what we have discussed that during the dark reactions, the carbon dioxide which is present in the environment is taken by the plant and it reaches to the chlorophyll, and where the enzyme system is fixing this CO2 to the useful form of sugar. So what we have discussed is that the CO2 is getting converted into the sugar, or I will say simple sugar, and that simple sugar is getting converted into other different forms of sugar such as starch or the polysaccharides like. Okay. So, this happens because the chlorophyll has the enzyme system known as the RUBP carboxylase oxidase. Okay. So, as the name suggests, the ribulose or the rubisco actually contains the activity which actually fix the carbon dioxide to form the sugar by doing the different simple steps, and all these simple steps are actually the part of the Calvin cycle. So, one of the crucial enzyme which is actually the rubisco or the RUBP carboxylase oxidase contains two different activities. It contains a carboxylase activity and it contains a oxidase activity. Okay. What is mean by the carboxylase activity is that it adds the carbon dioxide to the sugar. For example, in this case we have discussed already that the RUBP is getting converted into the by following several steps into the phosphoenol pyruvate, and that phosphoenol pyruvate then eventually get into different steps by the addition of the carbon dioxide. Okay. What is mean by the oxidase is that it actually oxidizes the substrate for and produces the carbon dioxide. So, in this case it will take up the sugar, it will add the O2 and it will produce the carbon dioxide, which means the rubisco is doing the two different activities, and these two different activities are actually doing exactly the opposite. For example, in this case the carboxylase activity are part of the anabolic activities, means the ultimately the sugar what is being synthesized is going to participate into the bodybuilding process, whereas the oxidase process is a part of the catabolism metabolism, means it is actually a breakdown of the sugar. Okay. So, these are two contradictory activities are present in a single enzyme. So, the question comes how and why this particular activity is present in this enzyme. First we will discuss about why this particular activity is present, and then we will discuss how the plant is utilizing this particular kind of activity for its own betterment or for utilizing it. So, if you remember that during the origin of life on the earth, the oxygen was not present, only the hydrogen was present and the carbon dioxide was present. And that is why if you recall the experiment of Steinmeier, where he has actually used the water, carbon dioxide and nitrogen and other kind of gases, and then he has produced the simple sugars and amino acids and all other kind of molecules. So, in that particular kind of primitive environment, when the photosynthetic organism appeared, they have actually appeared with the enzyme known as the rubisco. So, that time the rubisco was only getting exposed to the carbon dioxide. There was no oxygen. So, as a result, there is no oxygen in the primitive environment, and because of that the rubisco could not get a chance to optimize its active site. See, what happened is, during the evolution, any enzyme, if it is accepting one or the several substrates, eventually it optimizes its active site for doing one function and the other function. So, in an ideal situation, if the rubisco could have the evolutionary pressure of the carbon dioxide as well as the oxygen in the primitive environment, we could have seen the two enzymes, one which is this and the other enzyme which is actually this, means this is the carboxylase. So, we could have seen a rubisco derivatives which was only doing the carboxylase activity, but no oxidase activity, and we could have seen a rubisco form which is actually doing the oxidase activity, but not the carboxylase activity. But that is not happened, that never happened, because the oxygen was absent in the primitive environment, and that is how the rubisco was trained only to accept the carbon dioxide and to convert this carbon dioxide into the sugar, and during that process only the downstream reactions started producing the oxygen, or the light reaction itself has started producing the reaction. So, slowly the oxygen level gone up, but that was happened so late that rubisco has not get any chance to optimize its active site. So, what is the problem? The problem is that you are actually generating the sugar, and at the same time you are actually degrading a part of that sugar. This means you are doing by having these two activities at a same enzyme, we are actually doing the kind of a futile cycle, means we are actually activating, we are making some product and we are allowing it to break down. But in what condition? So, this is not the ideal condition in which the rubisco is going to show its oxidase activity. There are specific conditions in which the C3 plants are going to show photorespiration or the oxidase activity. So, because the rubisco can do oxidase as well as the carboxylase activity at the same time, and because this oxidase activity is degrading the sugar, or it is oxidizing actually the sugar, this that happens only when the light is present. This particular mode of oxidation is known as the photorespiration. Let us discuss what are the factors which really governing when the plant is trying to use the photorespiration pathway, and instead of doing the synthesis part, it actually doing the breakdown part. As we discussed, Rubisco is accepting carbon dioxide, it is also accepting the oxygen, and this is actually is the photo oxidation, okay, and this is the, anyway we know that. So, the first parameter could be that if you have a very low concentration of carbon dioxide to oxygen ratio, means if you have very low ratio of carbon dioxide to oxygen, means there is a optimal ratio, but if there is a high quantity of oxygen in the environment, that actually will drive the Rubisco to go for the photo oxidation pathway, means this degradation pathway. Number 2, if there is an increase in temperature, then the increase in temperature actually reduces the photosynthesis, okay. But at the same time, increase in temperature actually increases the photo oxidation. So, it reduces the photosynthesis, but it increases the photo oxidation. And increase in temperature also reduces. So, for example, in the case of aquatic plant, the increase in temperature also reduces the dissolved oxygen. So, it increase in temperature has a dual effect in terms of inhibiting the photosynthesis as well as activating the photo oxidation, as well as reducing or disturbing the ratio of carbon dioxide and oxygen which is dissolved in the water. Number 3, light intensity. So, light intensity, if it is a very high or if it is above optimum, that actually will going to induce, or that is actually going to force to the plant to go for the photo oxidation rather than the photosynthesis, because in those conditions there will be enormous production of ATP and NADH, and the photo oxidation pathway is going to utilize them. So, because of that, the photo oxidation is preferred over the photosynthesis. Number 4, age of leaves. So, you know that the young leaves will have the higher probability of different types of gases compared to the older leaves. That is why the photorespiration or the photo oxidation is going to be low in young leaves, but it is going to be high as the age of leaf. So, it is going to be very high in terms of the age of the leaf. So, let us see what are the different reactions which are happening when there is a when the Rubisco is utilizing its oxidase activity and doing the photorespiration. So, as you can see on the given slide, the 3 different organelles are being given, and the photo oxidation or the require the cooperation of 3 different organelles. One is chloroplast, the other one is called peroxisomes, and the third one is called the mitochondria. So, one of the objective of the the photooxidation is that it somehow trying to reutilize the oxidation byproduct, which are actually useless if they will remain in the chloroplast. So, for that purpose it actually joins hand with the other organelles, or it will take the help of other organelles and let the things to be in such a way that it should be utilized by the plant cell. So, that is why the photooxidation or the photorespiration occurs in 3 different organelles. One is chloroplast, other one is peroxisome, the third one is the mitochondria. So, first reaction is catalyzed by the Rubisco in a condition when the carbon dioxide and CO2 ratio is decreased, means there is a more quantity of oxygen in the environment. As a result, the ribulose 1,5 bisphosphate is directly, rather than getting carboxylated, it is getting oxidized by adding the oxygen, and it produces the 2 product. It produces 1 molecule of 3-phosphoglyceric acid and 1 molecule of 2-phosphoglycolic acid. The 3-phosphoglyceric acid can be converted into ribulose 1,5 bisphosphate following the reaction of C3 cycles. Okay. So, this is not a problem. 3-phosphoglyceric acid, if it is formed, it can be channelized back to the C3 cycle by following the Calvin cycle. But the 2-phosphoglycolic acid, the 2-phosphoglycolic acid is actually cannot be utilized as such until it is being converted into some other form, and that is why it is actually a useless product. So, 2-phosphoglycolic acid is first get converted into the glycolic acid, and that is done by a phosphatase, and so the 2-phosphoglycolic acid is being hydrolyzed and to generate the glycolic acid and the inorganic phosphate. The enzyme catalyzes this reaction is known as the phosphoglycolic acid phosphatase. As soon as the glycolic acid is formed, the glycolic acid is transported to the peroxisome, and the chloroplast is actually having a special transporter which actually transported the glycolic acid outside from the chloroplast, and then it enters into the peroxisome. So, as soon as the glycolic acid enter into the peroxisome, and if you cannot recall the peroxisome, peroxisomes are the organelle which actually are important for oxidizing the different types of molecules, and they actually take part in neutralizing the different types of toxic chemical as well. So, as soon as the glycolic acid enter into the peroxisome, it get converted into the glyoxylic acid, and this reaction so glycolic acid actually first undergone another round of oxidation by using the oxygen to form the glyoxylic acid and as well as the hydrogen peroxide. As soon as the hydrogen peroxide is formed, the peroxisome is full of catalases. So, that catalase actually converts the hydrogen peroxide into the water as well as the oxygen, and then the hydrogen peroxide then the glyoxylic acid is being taken into further. So, glyoxylic acid is then transaminated into a glycine, and then with the help of an enzyme known as transaminase. As soon as the glycine is formed, this glycine then moved to the mitochondria, the third organelle which is present, which is participating into the photorespiration pathway, and it enter into the mitochondria. Within the mitochondria, the glycine is getting converted into the serine, and in this process it produces a molecule of carbon dioxide, it produces the ammonia, and then it also produces the reducing power. So, the reaction which is going to be catalyzed at this step is that glycine, 2 molecules of glycine are taking up and adding with the alpha ketoglutaric acid, and then it is getting converted into the serine as well as the carbon dioxide and the glutamic acid, and 1 molecule of NADH is getting generated. So, once the serine is formed, the serine is again going back to the into the peroxisome, and once the serine enters into the peroxisome, it is getting converted into the hydroxypyruvic acid by a transaminase reaction, and in this process the enzyme hydroxypyruvic acid reductase, the transaminase through a transaminase reaction. So, hydroxypyruvic acid is formed. The hydroxypyruvic acid is then getting converted into the glyceric acid, and the enzyme which is catalyzing this reaction is known as the hydroxypyruvic acid reductase. In this step, the NADH is used, and as soon as the glyceric acid is formed, the glyceric acid then transported back to the chloroplast, and then this glyceric acid is getting converted into the 3-phosphoglyceric acid. So, the glyceric acid is getting phosphorylated with the help of an enzyme known as the glyceric kinase, and and as a result it actually forms the 3-phosphoglyceric acid, and that 3-phosphoglyceric acid is converted into the ribulose 1,5-bisphosphate following the usual reaction of C3 cycle, and that actually eventually complete or the regenerate the molecules to participate either into the carboxylase reaction or into the oxidase reaction depending on the conditions. So, these are the conditions. So, since the major this all is happening because the ribosco has a additional oxygenase activity, and the system has adopted itself to utilize even the oxidation product. But under these conditions there is a heavy losses as well as the several advantages to the plant, and let us discuss what are the different advantages as well as the disadvantages to the plant. Why the plant is adopting to the photorespirations. So, let us first discuss about the disadvantages. So, one of the most problematic disadvantage of photorespiration is actually waste lot of energy. So, it wastes the energy, as you might have seen that there are so many phosphorylation reaction where the ATP is used, there are so many reduction reactions where the NADH has been used. So, it actually wastes lot of reducing power in terms of ATP as well as in terms of the NADPH. So, that is the clear loss of ATP and loss of NADPH. Okay. So, it actually, as I said, these are the catabolic reactions, but there is no energy production in this. So, this is actually a photorespiration where the substrates are being oxidized by oxygen, but they do not get converted in terms of the oxygen. See, in the real respiration which actually happening in the mitochondria, there is a downstream electron transport chain, and that actually generates the large amount of ATP and NADH, whereas in this case there is no production of ATP or NADH. In this case there is a loss of ATP and NADH. So, that is actually a loss for the plant. The number 3 is because of this losses of food or losses of the sugar which is being produced, and the channelization of light energy into a waste process, the yield of the plant is getting reduced. So, because of the photorespiration, there is a low yield in the plant. Now, the question comes that if there are so many disadvantages, by following this simple mechanism, why the plant still have this particular kind of pathway exist? Why the plants are actually going through with this kind of pathway? So, the question to answer to this question is that there are several advantages as well as there are several achievements by going through a photorespiration process. What are these advantages? As we discussed, the photorespiration is happening in a condition when the light energy is very high. Okay. So, when the light energy is very high, the plant is producing the more number of reducing equivalents what it cannot used even for the downstream dark reactions or the biosynthetic reactions. So, under those conditions, these extra amount of ATP and NADPH are not good for the plant. So, or as well as in the chloroplast. So, in those conditions, these extra ATP and NADPH has to be lost, or it has to be because the photorespiration is happening because there is a low concentration of oxygen, there is a high concentration of oxygen and there is a low concentration of carbon dioxide, and on the other hand there is a high light intensity. And so, one of the thing is that it dissipates the reducing equivalents, so that there will be no feedback mechanism within the light reaction, and because of that there should be no complete blockage or complete blockage of the photosynthesis. So, it actually start wasting the extra ATP and NADPH which are being produced by the light reaction, so that these ATP and NADPH should not go back and say that we do not need any more ATP and NADPH, because if you remember, the ATP is being formed by ATP and PI. Okay. Now, this ATP, if you have very high quantity of this ATP, there is no way that you can generate more number of ATPs, okay, because there will be no ADP and PI available. That is why you have to keep running some system, so that ATP is getting break down into ADP and PI, and so that you will have the substrate, because this is happening in the presence of sun. Okay. So, sunlight is coming and it is trying to that electron photoelectron transport chain is running, and that actually is that need this ADP and PI, and that will only come if there is a photorespiration. Same is true for the NADPH. Number 2, similar to or closer to this logic, that if there is a very high light quantity, that actually will produce very high quantity of these reducing equivalent, and these and on the other hand, because there is a high photoelectron transport chain is running, by running the photorespiration, it protects the plant from getting the photo injury or photo inactivation. There is a classical example where the photorespiration, why the photorespiration is protecting a plant from getting damaged. So, there are Arabidopsis thaliana mutants, and people are generating the several types of mutant in the plant, and in this particular mutant, this mutant does not survive in and so this mutant is actually does not be able to do photo respiration, and so under those conditions this mutant is very sensitive for environmental conditions, or it cannot grow in a normal environmental conditions where the oxygen percentage is 21% and the carbon dioxide is only 0.03% or 0.3%. So, it cannot grow in a normal environmental conditions, but it grows into a condition where the carbon dioxide percentage has to be raised to 2%, and that happened because this particular mutant of Arabidopsis cannot perform the photorespiration, because in this particular mutant it is very susceptible for the high quantity of oxygen. So, that actually in nutshell, the photorespiration is bad in terms of as far as the yield or the production of the plant product is concerned, but as far as the plant physiology is concerned, the photorespiration is a adaptation in the plant to protect the plant from the light energy or the light injury into the system. So, it actually wastes so much reducing equivalent, so that the photosynthesis keep continuing. So, photorespiration is reducing the is happening because photorespiration is happening because the Rubisco has dual activity. It has a carboxylase activity as well as the oxidase activity. So, what is the solution to this problem? What is the solution to this problem is that as we discussed, right, photorespiration is happening only because because there is a very low concentration of carbon dioxide and there is a very high concentration of oxygen. So, under those conditions the Rubisco is also accepting the oxygen and doing the oxidation process. So, the solution to this problem is that if you split or if you do a better carbon assimilation or carbon storage from the environment, and if you do not use the Rubisco to capture the carbon dioxide from the environment, and in that process you are going to cut down the loss pathway. Means, see, Rubisco, as we discussed many times, this carboxylase pathway and as well as the oxygen pathway, this is actually a loss making pathway, right. So, if we remove this with any other enzyme system, it actually can help to have the better CO2 sequestration, and better CO2 sequestration as well as this problem is going to be removed, and that is what is happening in a separate class of plant. Those plants are called as Sifo plants. So, what is the solution to this problem? The solution to this problem is that we have to have a better CO2 fixation mechanisms. So, a better CO2 fixation mechanism can be done by going to the by adapting the two mechanisms. One, so how to get a better CO2 fixation? We to have a better reaction vessel. What is mean by better CO2 better reaction vessel? See, the carbon fixation is happening in a plant or in a leaf actually. Okay. So, if we have the leaf anatomy in such a way that the carbon dioxide what you trap will remain within the leaf, so that the local concentration of carbon dioxide is more, and that you can do by adopting to a different types of leaf anatomy. Okay. So, what is that leaf anatomy that we will discuss anyway. The number 2 is a better CO2 fixation system. A better CO2 fixation system means that instead of Rubisco, if you have a CO2 fixation system which actually can should not have the oxidation activity, number 1, and number 2, it should have a it should do a very efficient CO2 fixation into the carboxylated product, and these carboxylated product can be used to produce the sugar. So, let us discuss about the better reaction vessel. So, in the better reaction vessel, as I said, it is all about the leaf anatomy. So, as you can see, so what you have in the leaf? You have in the, if you take a transverse section of a leaf, what you have? You have a layer of epidermis. So, what is mean by the better reaction vessel is that you can have a better leaf anatomy. So, in a better leaf anatomy, you can have a CO2 fixation system very deeply integrated into the system, so that the whatever the CO2 you absorb or whatever the CO2 you should sequester in this kind of structure should maintain a very high CO2 concentration, number 1, and number 2, it should have take more longer of time to escape from the system. So, if you see a leaf anatomy of a leaf from the Sifo plant, what you can see is the top and bottom, these are the so this is a TS section. So, what you can see is the top and bottom, there are epidermis, and then there are series of mesophyll cells. These mesophyll cells are actually the cells which actually contain the chlorophyll, and they are actually participating actively in capturing the light energy and as well as running the downstream dark reactions. What you have is in the center, what you have is the xylem and phloem, and then surrounding the xylem and phloem you have the bundle sheath cells. These bundle sheath cells are also participating in the photosynthesis. Okay. So, you can see that the carbon dioxide, and these are the stomata which are actually the opening on the leaf, so that the gas or the air enters through this pore, either the carbon dioxide or the oxygen, and then it reaches to the first layer of mesophyll cells, and then they will catalyze the metabolic reactions, and then eventually these cells which are present in the bundle sheath and the cells which are present in the mesophyll are going to cooperate with each other, and it is going to give you the better yield as well as the lower chances of going through the photorespiration. Apart from this, why this particular kind of anatomy is favoring towards the photosynthesis rather than the photorespiration is that because there is a thick layer of mesophyll and then there is a bundle sheath, it also reduces the light intensity which is actually going through the leaf. So, let us see what are the different reactions which are happening while the system is following a better fixation system and the better reactions, and this another class of CO2 fixation is known as the C4 cycle, or and the plants which contains this kind of anatomy is known as the C4 plants. So, as you can see on the given PowerPoint slide, the CO2 fixation in the C4 plant actually requires two different compartment within the leaf. One is called the mesophyll cells, the other one is called as the bundle sheath cells. And in this case, the CO2 fixation is not being done by the CO2 fixation is done by two times. One is when it is done in the mesophyll cells, the other time when it is done in the bundle sheath cells. And because there is a two times CO2 fixation, the yield of the yield from these kind of C4 plant is very high compared to the C3 plants. So, in this there are four steps in the complete process of CO2 fixation. So, there is the initial fixation, and as we discussed in C4 plant, the fixation is happening twice. One is in the mesophyll cells, the other one is in the bundle sheath cells. So, in the initial fixation, the primary molecule which is used to fix CO2 is the phosphoenol pyruvate. So, phosphoenol pyruvate actually take up the carbon dioxide which is dissolved in water. So, these phosphoenol pyruvate does not take the carbon dioxide which is present in the gaseous form, it actually take up the carbon dioxide which is dissolved in water and it is present in the form of bicarbonate. So, the phosphoenol pyruvate get carboxylated and that is how it fixes the carbon dioxide into the oxaloacetic acid, and the enzyme which actually catalyzes this reaction is known as the phosphoenol pyruvate carboxylase. This enzyme is very unique actually, because this enzyme does not work optimally only in the range of 30 to 35 degrees Celsius, and as a result of this carboxylation, the mesophyll cells actually produces the oxaloacetic acid. This oxaloacetic acid is then being modified to in two different ways. In one way, the oxaloacetic acid is being transaminated to form the aspartate, and then the aspartate and in this the enzyme is known as the transaminase. So, the oxaloacetic acid can be transaminated in two different ways. One, where the oxaloacetic acid oxaloacetic acid is combining with the glutamic acid with the help of enzyme known as transaminase, and then it generates the aspartic acid and as well as the alpha glutaric acid. In the other class of transaminase reaction, oxaloacetic acid combined with the alanine and with the help of transaminase, then it produces the aspartic acid as well as the pyruvate. Nevertheless, either of these pathways, the aspartic acid then transported out of the mesophyll cells and they utilize the plasmodesmata to go to the bundle sheath. Once they reach to the bundle sheath cells, then again the aspartate is getting into the transamination and then they will form the oxaloacetic acid. On the other hand, the malic acid is being transported directly into the bundle sheath cells. On the other hand, the oxaloacetic acid is being converted into the malic acid, and the enzyme which is being catalyzing this reaction is known as the Malic dehydrogenase. In this pathway, the first molecule of NADPH is being used. This NADPH which is being produced during the photoelectron transport chain, and both the malic acid as well as the aspartic acid then transported to the bundle sheath cell. So, that is the second step where the carboxylated products from the mesophyll cell are transported to the bundle sheath cell through plasmodesmata. Plasmodesmata are actually the junctions between the two cells, and it is being used for providing the transport between the two plant cells. oxaloacetic acid cannot be transported as such because it requires the special transporter, and that's why the oxaloacetic acid is being converted into the aspartic acid as well as the malic acid, because the plasmodesmator allows the transport of these two molecules. Once the malic acid is entering into the bundle sheath cells, then there is a second round of fixation which is going to happen. So, the first fixation we have seen that carbon fixation happens when the phosphoenol pyruvate has taken up the carbon dioxide from the environment and produces the oxaloacetic acid, and now once the malic acid is is transported to the bundle sheath cells, these malic acid then releases. So, is going to have the second round of CO2 fixation. In the second round of CO2 fixation, the malic acid is then going for the release of carbon dioxide. So, malic acid is getting converted into the pyruvic acid, and in this process the CO2 is released and CO2 is released. So, in the first reaction the malic acid is then converted into the pyruvate and CO2 is being released. This reaction is catalyzed by a Malic enzyme, and so in this reaction a molecule of NADH is being produced. So, the malic acid is getting converted into the pyruvate with the help of enzyme known as malic enzyme, and the carbon dioxide is produced, and the one molecule of NAD plus is being converted into the NADH. As soon as the pyruvic acid is formed, this pyruvic acid is transported back to the mesophyll cell, and there it is going to get converted into the phosphoenol pyruvate. The CO2 released, so CO2 is also being released from the malic acid. This CO2 which is released from the malic acid is then being taken up by the ribulose 1,5 bisphosphate, and then it converts and produces the and goes into the Calvin cycle. On the other hand, in the second reaction the aspartic acid is transaminated to form the oxaloacetic acid or alanine. So, in the first reaction oxaloacetic acid is to form, so aspartic acid is then transaminated to form the glutamic acid and oxaloacetic acid. The enzyme catalyzes this reaction is known as the transaminase. Ultimately, the oxaloacetic acid is then converted into the phosphoenol pyruvate, and it releases the carbon dioxide, and this carbon is also being used for running the Calvin cycle in the bundle sheath cells. So, phosphoenol pyruvate and the enzyme which catalyzes this conversion of oxaloacetic acid to phosphoenol pyruvate is known as the phosphoenol pyruvate carboxykinase. In this process, one molecule of ATP is used, and the CO2 which is being released from this is then used in the Calvin cycle. The third step is the carbon assimilation. In the carbon assimilation, the bundle sheath cells, the CO2 whatever is released either from the malic acid or the aspartic acid is used in the Calvin cycle. The only difference is that the phosphoglyceric acid which is formed in the Calvin cycle is not being further degraded into other product, and this phosphoglyceric acid is being transported to the mesophyll cells to get converted into the glyceraldehyde 3-phosphate, and subsequently once it gets converted into the triose phosphate, then only it is returning back to the bundle sheath cells, and then it completes the Calvin cycle. At the end what we have done, we have generated the phosphoenol pyruvate in the bundle sheath cells, and in the other hand the malic acid has produced the pyruvate. So, now if you want to run this complete C4 cycle, the last stage is to regenerate the phosphoenol pyruvate in the mesophyll cells. So, phosphoenol pyruvate is being generated from the pyruvate which is being produced in the bundle sheath cells. So, the after the transamination, the alanine is produced in the case of aspartic acid, and in the case of malic acid the pyruvate is produced. So, the alanine is transported back to the mesophyll cell, and pyruvate which is being produced by the malic acid is then phosphorylated, to form the phosphoenol pyruvate. So, there are two rounds of phosphorylation, and in this the ATP is used, and the phosphopyruvate is pyrophosphate is released, and the phosphoenol pyruvate is formed. The enzyme which is used in this reaction is known as the phosphopyruvate dikinase. This enzyme is actually a cold labile enzyme, and it does not work if the temperature is above 12 degree. So, ultimately what we have done, we have generated the phosphoenol pyruvate back, so that the reaction is going to be over, and this phosphoenol pyruvate is then again being used to catalyze the CO2 fixation as well as and continue the cycle in both the different types of cells present in the leaf. So, this is a usual adaptation mechanism in the Sifo plant, so that the photosynthesis is going to be more efficient, as well as the photosynthesis is so in this cycle the production is going to be more optimal, because the Sifo plant will not waste the energy or as well as into the sugar into the photorespiration process, and they will avoid that because they have avoided the rubisco as the primary carbon CO2 acceptor, and because they are using the PEP carboxylase as an enzyme. So, PEP carboxylase does not have the oxidase activity, and because of that the initial CO2 sequestration from the environment, because as you know that rubisco is being affected by the ratio of carbon dioxide as well as the oxygen. So, because the rubisco is directly taking the carbon dioxide from the environment, and that is how the oxidase activity is coming into the picture. Whereas, in this case, although we are giving the carbon dioxide again back to the rubisco only to catalyze the CO2 fixation, but at that point there is no oxygen present in the environment. This carbon dioxide is coming by the breakdown of malic enzyme or malic acid actually, or in the other case it is coming because there is a breakdown of oxaloacetic acid. And because of this kind of unique chemical because of this unique anatomy, as well as because of this unique chemical reaction or chemical mode of CO2 fixation, the CO2 fixation in Sifo plant is much more efficient. So, with this we have discussed about the different aspects of photosynthesis, and now in a subsequent class we are also going to discuss about the different kinds of factors which are going to play crucial role in regulating the photosynthetic activity, and ultimately we are also going to discuss the different factors which are actually playing role in increasing the yield from the plant. So, with this we are going to conclude our lecture here. Thank you.